So what I'm going to tell you about is some of the things we're doing with QIIME, which is this package that stands for Quantitative Insights into Microbial Ecology. And so, so a lot of the emphasis on QIIME is how do you get from your sequence data to a visualization that you can actually use. And so this project was initiated in my lab by Greg Capareso, who's here, Justin Kaczynski, who's now at Second Genome, and Jess Dombau, who are now at Thermo Fisher, and a bunch of people in my lab and collaborators around the country and around the world contributing to this open source effort. So, so the backdrop to all this is familiar to many, perhaps all of you by this point, is that sequencing is getting dramatically cheaper, so much so that when Steve Quake sequenced his own genome at the end of 2009, it was literally almost 1 million times cheaper than at the completion of the Human Genome Project in 2001, and it's another couple of orders of magnitude cheaper since then. So as a result, we really have this democratization of sequencing, right? It used to be that a small trickle of sequences occasionally escaped from behind the walls of a few centralized sequencing centers. But now you really have this democratization of sequencing where in every department and soon in every lab, you'll be able to do the sequencing yourself. And it's relatively likely that sequencing instruments are going to go the way of, say, PCR machines, where a couple of decades ago they were very expensive, but now many of you probably don't even know how many PCR machines you have in the lab, right? And so sequencing is going the same way with a huge democratization of the techniques. But then the issue is that you run into this problem, which I think is also familiar to a fair number of you, and is a backdrop to this workshop. That interpreting all the sequence data in the phylogenetic trees is really hard. So just to illustrate this, I'm showing you the first 9 out of 130,000 sequences from a recent project we did with Noah Ferrer's group. Here's the first 0.1% of the tree file, and here's what happens if you just do a naive visualization of the tree. And you can really tell that you've got your work cut out for you understanding this if you try to view the data in this form. And so it doesn't even help you if I tell you the study design, right? So what we were doing was a pretty basic biogeography project where Noah's initial idea was to look at things like, does the space bar have more kinds of microbes on it simply because it's larger than the other keys? You know, the keys are desert relative to the lush valleys of your fingerprints where maybe fewer microbes would survive there. And is there a biogeographical dividing line between the G key and the H key, parallel to things like the Wallace line that divides the Asian fauna from the Australian fauna? So anyway, another view of the same data I just showed you makes the main things going on in the system immediately clear, right? So now each point is a microbial community, and you've seen a lot of these kinds of plots before in the morning session. What these are a principal coordinates analysis where you measure the distance between each sample, and then you try to lay it out in 2 or 3 dimensions to maximize the amount of the variability in the data that you can show in those small numbers of dimensions. And anyway, when we use this visualization technique, you can see that all of the fingertips and all the keys from the first person, the second person, and the third person form these 3 very nice, very discrete clusters. If instead I color everything according to what's a key and what's a fingertip, you can see they're essentially indistinguishable, right? So this shows very clearly that the main thing going on is that the microbiome is very personal in nature, and we transfer our own microbes from our fingertips to our keyboards as we type. So, so this came out in PNAS a couple of years ago, but more importantly, it was on CSI Miami, so you really know it's true. Anyway, so I'm gonna skip over this section on how we do the microbial sequencing, because I think everyone here is familiar with this idea of using direct environmental sequencing as pioneered initially by Norm Pace, who we have the honor of having here in the room, to take environmental samples, amplify specific marker genes, sequence them, but now instead of blasting them and looking at what's in the database already, in order to avoid the problem that you're going to match other uncultured microbial sequences, we advocate taking this explicitly phylogenetic approach of doing multiple sequence alignments, building phylogenetic trees, and then relating the sequences on those trees back to the samples that they came from, and also crucially back to their closest relatives, so that we can understand things about the biochemistry and the genetics of the closest cultured bacteria to the unculturable bacteria that we see out in the environment. But this runs us into another big problem, which is that these large trees can be really hard to understand and to analyze. So this is from a paper we did in 2005 with Jeff Gordon's lab, where we were looking at 5,000 sequences out of the mouse gut and 12,000 sequences out of the human colon, coloured in red and blue respectively. And you can kind of see some red clusters and some blue clusters on the tree. And at the time, there were some statistical techniques that would let us determine that those two groups of taxa were not identical to one another. But there wasn't any phylogenetic metric that we could use to tell how far apart the samples were. And so, so this might not seem so bad with only 2 colours, but what I haven't told you yet is that these were from 21 different human samples and from 19 different mice. By the time you paint that tree in 40 different colours, and you're trying to figure out is turquoise more similar to cyan or to aqua, right? It's really hard to figure that out visually. And more generally, there was this period, and I don't mean to pick on this paper specifically. It's just the first example I pulled out of the stack on my desk when I was putting these slides together, where what you have is an exploration of a very interesting new environment, and in this case the termite gut. And what you want to do is you want to figure out what's new about the microbes in those communities. So you're reading the paper and you come along to Figure 3. Which is a phylogenetic tree with the new sequences inserted into the rest of the tree. And then you come to Figure 4, which is another tree, and Figure 5, and Figure 6, and a whole lot of additional figures with the tragic inevitable conclusion, right? So what we have to do is we have to somehow escape this issue of having all this tree data that we can't interpret. And the solution to this problem really came from the PhD thesis work of Kathy Lozapone, who graduated from my lab a couple of years ago. And he used this very simple definition of distance on a phylogenetic tree as a fraction of the branch lengths on the tree unique to any particular environment. So Mitch told you a little about different distance metrics you can use to compare communities. The advantage of the unifrac metric is that what it lets you do is it lets you tell how closely related or distantly related 2 communities are using all of the evolutionary history of the microbes in those communities. And so the intuition here is that if you have 2 unrelated communities, all of the branch length is going to lead only to members of the one community or only to members of the other, but never to both. So those are as unrelated as possible, and we define that distance as 1. Whereas in contrast, if you have 2 identical communities, everything, and you have complete sampling, everything you find in the first community, you also find in the second. So all of the branches on the tree are shared, shown here in purple. None of them are unique, and we define that distance as 0. And then any actual pair of communities, you'll have some unique branches and some shared branches. And so this is a unique fraction or unifrac metric, which has now become fairly popular for these kinds of studies. And so what's great about this is that we can use the universal tree of life as a measuring stick to combine communities from anywhere. So if we take that one tree of life and look at red, yellow, and blue communities, what we can do is we can compare the red to the yellow community, the red to the blue, and the yellow to the blue. Summarize all those distances in a distance matrix, and then as soon as we have that matrix, we can use standard multivariate techniques like principal coordinates analysis, hierarchical clustering. You can also do all kinds of other things on the underlying data for this, like machine learning, to try to figure out what taxa you have unique to a particular environment. You can do source tracking, and you can do a fairly large number of other analyses. So having these tools in hand, Kathy initially decided to look at everything. And so she went to GenBank and put together a meta-analysis of over 100 studies, over 200 samples, and over 20,000 sequences, which again at the time was an absolutely massive data set, right? And so what made this possible was that she had 16S ribosomal RNA gene sequence data for all of those different studies, which made it possible to combine them. So fingerprinting techniques like DGGE or T-RIFLET or so on are very useful within one particular study system, but very difficult to combine across different studies. By having the DNA sequence data mapped to the same phylogenetic marker, we're able to do those combinations. And so, so she looked at a set of samples literally covering the world, everything from extremes of temperature, pH, to just about every other physical or chemical factor you could imagine. And the question was, out of all this complexity, would we see any clear patterns relating all of these dissimilar samples? And much to my surprise, the clear answer was yes. We saw this very distinct split between the saline and the non-saline samples. You'll see a few samples in blue in the middle. What those are the estuaries, where you really do have a mixture between the saline and the non-saline. And this ability to integrate over all these different kinds of data is precisely what we need for the built environment, where we have all kinds of different materials, all kinds of different conditions, all kinds of different sources that the microbes might potentially be coming from. And so this is really the precursor to the kinds of data integration efforts that we're trying to do now. However, doing this meta-analysis was a complete disaster in terms of metadata. So you'll notice that these are very broad categories. Initially, Kathy was planning to enter the actual pH, the actual temperature, a whole lot of other variables for each environment, but it's essentially impossible to get these out of the literature after the fact, and the amount of work involved is tremendous, which is why we place such an emphasis on getting the metadata into a consistent format so that you can do these kinds of things with the click of a button, instead of, as Kathy did, spending several years reading 400 different papers to find 110 of them that actually had useful information. And I'll point out that the method of measuring these differences really matters a lot. So here's a view using slightly newer, slightly expanded data set on the same data I showed you before with Unifrac, with the saline, non-saline split and the estuaries in the middle. If instead you take the taxon table and you use a dissimilarity measure that takes OTUs or other taxa and just asks what is the overlap in these taxa across the different samples, you apply it to exactly the same data. Because most microbes are not in most environments, and because the data matrices are very sparse, you tend to get these spike out effects at 90 degrees relative to one another. So with the Kaczynski quantitative symmetric metric and Euclidean distance, for example, and you lose the obvious patterns that you're able to see with the phylogenetic metric. So how you measure distances really has a large impact on your analysis. Anyway, one thing that was surprising to us in this was that what we thought were going to be extreme environments weren't really outliers. So if we look at sediments from hydrothermal vents or from hot springs in Yellowstone, they just look like the other saline and non-saline communities respectively. Although by collaborating with Rick Lay and Jeff Gordon, Kathy was able to find some really extreme environments. And when we add these to the plot, what happens is we squash up all of these samples from all these physical environments just into one side of the graph. We introduce this new axis that explains twice as much of the variation as the saline, non-saline split I showed you. But it turns out you don't have to go very far to find these extreme environments. They're right here within us. So mostly what we're seeing is this profound split with the vertebrate gut, which is very distinct from everything else. And then other human-associated samples are intermediate. And again, this speaks to the value of being able to combine host-associated samples with environmental samples and get a picture globally of how these different kinds of microbial assemblages distribute over the environment. So what really enabled the jump to next-gen sequencing, which Mitch was doing at about the same time and really pioneered in two very nice papers, one in PNAS and one in Science, looking at microbial ecosystems, was this observation that you didn't necessarily need the complete 16S ribosomal RNA in order to be able to do this community clustering. So it turns out that if you just take a short fragment of the sequence and starting from one of the well-characterized primers and go forward 100, 150, 200, or 250 bases, depending on which region you pick, you might get essentially the same clustering pattern. And so basically, to cut a long story short, if you pick a good region of the 16S, so these are the full-length sequences for the humans and the mice I showed you before. And these are from the Guerrero Negro microbial mat, one of the systems that Norm studies, which is the most diverse microbial community that we know of on Earth. And if you pick a good 200 base fragment of the sequence, you get exactly the same clustering pattern right down to the same outliers. Now it's important to note that you could also pick a bad region of the sequence, and in this case we see some of the humans looking more like the mice than they look like the other humans. And at first you could be very excited about this, right? Like you might go back to your clinical data and ask, what is it about these people that makes them resemble the mice? Are they vegetarian? Do they live in a confined space? Are they very tiny or what, right? And then you might be sad later when you got the full-length sequences and you found out that the result just wasn't true, right? But the good news is that the regions of the 16S you can use to get the same community clustering pattern are by now pretty well known. And in a lot of those regions, even the short fragments you get that you can use on Illumina sequencing are perfectly fine for this kind of community analysis. Although you should be aware that they're less good if you want to identify specific pathogens or specific taxa that you're trying to discriminate from one another. So they're very good at this whole community level, even with very short reads, but you'll need longer reads if you want to identify a specific organism that you care about. So built on top of Unifrac, we put together this overall pipeline called QIIME, Quantitative Insights into Microbial Ecology, And the goal with this was to combine all of the steps for integrated analysis of hundreds of samples. So the strategy here was to use this barcoding approach, where we use error-correcting codes to tag 16 S genes from potentially hundreds or now even thousands of samples, using a DNA barcode that we introduce at the PCR step. And then a lot of what we've done is mixing the samples together and sequencing them on 454. Although, like many other people here, we have been switching to Illumina, and Greg and Jack and others here have done a lot on developing those protocols and making it possible to use QIIME on Illumina data, which is a whole additional scale of sequence acquisition from 454. So a 454 run might yield 1 million sequences if you're lucky. An Illumina run, if you're using a full flow cell on the HiSeq, can yield as many as 1 billion sequences. So then, because each sequence has a barcode, we can tell what sample it came from. Then we can clip off the barcodes, align them and identify the taxon-specific regions, use the sequences to build phylogenetic trees, and then use the trees to cluster the samples like I showed you. And so the goal behind QIIME is to resolve a lot of the technical challenges with next-gen sequencing data, primarily by providing a pipeline where some of the steps are things that are new that we develop ourselves, but many of the steps are integrating existing tools and making it easier to use them so that you don't have to track down 100 different programs on the internet and install each of them on your computer. So basically, starting from the output of the sequencing machine and from the metadata, which I will talk more about tomorrow, we do pre-processing, including primer removal, demultiplexing, quality filtering. You can do denoising, although depending on your analysis, you might or might not need to do that. So for alpha diversity numbers, where you care about absolute estimates of, say, how many species were in the sample, you care a lot about denoising. But if you care about overall patterns of community similarity, it's not so important. It has had SRA submission in the past, but one thing you should be aware of is that the SRA, the NCBI's short read archive, is in general not accepting new submissions of 16S data. There's only a few specific things that they're continuing to support. So what we're doing now, especially to get the metadata consistent, is through the QIIME database, you're able to export the sequences along with their associated metadata to ENA, the European Nucleotide Archive. And that's part of INSDC, the International Nucleotide Sequence Database Collaboration, which is a set of public databases that's a permanent repository for all kinds of sequence data. So then the next step is OTU picking, taxonomy assignment, and building an OTU table. And then from that, the sequence alignment and the phylogenetic tree. And for each of these, there are many different algorithms that you have a choice of. From the OTU table and the phylogenetic tree, you can finally start to do analyses and visualization. So things like alpha diversity to count how many species are there, things like beta diversity to look at the patterns of similarities and differences among your communities. And then a whole lot of visualization approaches, including things like the principal coordinates plots you've seen a lot of, histograms of the distances, OTU networks, which I won't talk about here, different kinds of clustering and so forth. Incidentally, a lot of people. Yes, so I have some slides on the stuff that I'll mostly skip through. The error correcting codes and the barcodes are really useful for minimizing the error rate of misassigning a sequence to the wrong sample, and this can be really important. This can be really important if you want to make sure that you don't have, say, a particular pathogen accidentally being detected by cross-contamination. There's several different denoising algorithms in there, including some that we've developed ourselves. So this is one by Jens Rieder, who was in the lab until 2011. We also recently added Acacia, which is a new technique from Phil Hugenholtz. You can do chimera checking. So chimeras are something you should be aware of. They're incomplete template extension. Can basically give you a hybrid sequence where it starts with one sequence and then it picks up with the end of another sequence. And if your PCR conditions are not very good, there have been reports that as many as 50% of the sequences in your PCR reaction can be these kinds of chimeras. However, I think it's fair to say that the techniques for detecting chimeras are still in their infancy. So what we have here is the same set of sequences run through 3 chimera checking packages, QIIME, Chimera Slayer, and Perseus. And as you can see, the overlap between these different algorithms is very low. there are far more sequences that are called as chimeric by only one algorithm, or by two of the algorithms, and by all three together. And so as a result, in order to get rid of all the chimeras, you're often losing so many sequences that are not chimeras that you would have been better off keeping the sequences and accepting the error rate depending on what analysis you're doing. So again, chimeras are very important for estimates of the number of species. They're less important for things like machine learning, where you care more about having a high count of sequences. Let's see, there's a bunch of OTU picking methods, including this really nice reference-based protocol that Greg put together, where basically the idea is to recruit to a reference tree and then pick de novo the additional clusters for just the sequences that you can't already get mapped into your reference OTU space, and so forth. Anyway, a lot of people are still puzzled about why the technology is called 454, and this explanation comes from my collaborator Rick Bushman at Penn, who we did some of our first 454 sequencing with, and he assures me that Fahrenheit 454 is the temperature your money burns at when you get into next-gen sequencing, right? So the cost of sequencing is dropping all the time, but you have to do a lot of it at once, so it's still relatively expensive to do each run. On the other hand, the first time we did this at the end of 2007, it cost us $12,000, and we got back 500,000 sequences. And at the time, the Sanger facility down the hall from my lab was still charging $8 a read. So if we'd done it, if we'd continued to use the Sanger technology, it would have cost $4 million instead of 12,000, and they wouldn't be finished yet. So I wouldn't be able to show you the rest of the stuff. So I'm going to skip over that in the interest of showing you some of the things that you can do with QIIME and some of the other tools that we're developing in the lab. So one thing we did relatively early on, and this was the work of Liz Costello, who's now working with Dave Relman at Stanford, and this was in collaboration with Noah's group and Jeff Gordon's, is putting together an overall map of the human-associated microbes. And you've seen this data set reused in a couple of the talks this morning, which is exactly what we had in mind for it. But basically what you can see is this very distinct cluster for the mouth, a very distinct cluster for the gut, and then the skin is a lot more spread out and a lot more diverse. So if you're using one skin site as a proxy for what human-associated microbes look like, that's actually not such a good idea. So what these pie charts represent, I don't expect you to read the taxa, but just notice that the colors in different places are completely different. So the skin on different parts of the body has radically different microbial communities, and so you probably want to look at multiple sites on the same person in order to get a good picture of what's on the skin. Anyway, in this study, so what was nice about this is that we had a temporal component to it, so we could see how the microbes were changing over time. And so on the y-axis here we have unifrac distance, where so a larger bar means more different. And so two samples from the same body habitat are less different than two samples from different habitats. Within a habitat, two from the same person are less different than two from different people. And then within a person and a habitat, two samples collected a day apart are less different than two samples collected 3 months apart. And then the mouth was by far the most stable habitat we looked at over a span of 3 months. The skin the least stable, and then the gut was in between. Actually, one thing I should say about this slide, both in studies of the hand, and this is work we did with Noah Ferrer, and in the gut, and this is work we did with Jeff Gordon, what you see when you do OTU level analysis is that there's extremely little that's shared between different individual people. So in the case of the undergraduate hand project, we see about 2,400 OTUs that were found on at least 2 hands, so we're reasonably sure that there are not sequencing error and so forth. Out of those, only about 750 were on 2 hands, about 450 on 3, about 300 on 4. You can see how rapidly this tails off, so there was essentially nothing that was on more than about a third of the hands that we looked at. And you see exactly the same pattern in the gut. So again, taking one human-associated sample and using it as your reference for what a human sample looks like is not a very good idea, just because of this huge and unexpected diversity between different people. And this diversity result's also been confirmed in the Human Microbiome project, where for any taxon you pick and for any body site, and this is from shotgun metagenomic data rather than 16S sequencing, for any taxon you pick, so for any taxon you choose, so this is Bacteroides, for example, that someone has a lot of, you can find someone else who has hardly any of it. And so this diversity is a very consistent pattern. We still don't know the sources of this diversity, but we do know that different individuals are really different from one another. But one thing that's frustrating about this is that a handful of time points isn't really sufficient. So in our project we looked at 3 time points. In the HMP, sorry, in our project we looked at 4 time points, and in the HMP we only looked at a maximum of 3 time points per subject. And to illustrate why this is limiting, so this is QIIME, which is the viewer we use at the moment for QIIME output. So there's the gut down there, there's the mouth over there. Oh yeah, and one thing a lot of people don't know is that with this sort of thing you can, with QIIME you can recolor the stuff really easily. So if I want the mouth to be a different color, I can just change that and update it and so on, right? And so a lot of these tools have features that are maybe not so well documented and that you might not have encountered, but they make your life a lot easier when you're doing these kinds of analyses. But anyway, if I take this and I rotate it around to give you an explicit axis for time, which is so with time you can take any continuous variable metadata category, like time or like an axis or light intensity or so on, and plot it as an additional axis. With just 4 time points, so these are 2 groups of 2 a day apart, you can't see the 1 day interval. You can kind of see the 3-month interval, but it's very difficult to get at dynamics, right? What you'd really like to do is you'd really like to go back to some of the same people and sample them every single day for like a year and a half. So with the Illumina technology, it was finally cheap enough to do that, and it looks like this. And you can see that the concordance between the 454 data and the Illumina data is really nice. So what this is telling us is that the effect size of the difference in primers and the difference in sequencing technology is smaller than the effect size of the differences between different people, right? And so there's a lot of sources of technical variability that might affect your result. However, what you need to know is the effect size larger or smaller than the biological thing that you're trying to find out? And anyway, so having these very dense time series, we can finally look at dynamics. And so Antonio, he's here at the meeting, put together this very this movie using as a framework all of the data from the Costello et al. Study, and then animating the two individual people. And so each step in this video is 1 day in the life of the microbiome. So the palm samples from 2 people, the mouth and the gut. And you can just immediately see the variability. Like how variable the skin is compared to the mouth, for example. You can also immediately figure out. So you can also see things like how the individuals preserve their identity, despite the fact that they cohabit and have all kinds of opportunities to exchange microbes. And you can also see how valuable this kind of visualization technique is going to be for things like the built environment, looking at how where you do a very detailed time course, and you see how the building changes over the course of the day or over seasonal variation and so forth. And I should also mention that, that to do this, which was one of our first large-scale Illumina sequencing efforts, Greg and others in the group had to do a huge number of changes to QIIME in order to be able to process the much larger data sets that we were now getting. So you can also do a bunch of stuff with increased spatial resolution. And so this is Chris Lauber, one of the postdocs currently working in my lab in NOAA's. And so he was interested in the microbes on the human face. And so he began with his own face, which he divided up into 50 different patches. And when you just do the PKA plot, it's kind of hard to interpret. So you have 36 and 37 being different. Those are the lips, and like I showed you, the oral community is really different. And you can kind of see some patterning in the colors here, which are numbered sequentially by what point they are. But it's kind of hard to see from this kind of abstract space, right? What you'd really like to do is you'd really like to just draw a map of your study site, whether it's your face or whether it's a house or a city or the continent of North America, and just paint the information on there directly. So Antonio developed a software called SitePainter that lets you do exactly that. So for example, if I color the face by PC1, you can immediately see the left-right symmetry, which you really couldn't see in that plot I showed you before. If I show you PC2, it's a bit puzzling because there's one patch that's really different. Our current conjecture about this is that it's because our subject wears glasses and tends to adjust them from time to time, and your fingertip communities are pretty different from what's on your face. And then if we look at PC3, what you see is the separation between the lips and the rest. But you might also want to do things like paint on say taxonomic information and relate it to the names of things that you know and care about. So if I show you Propionibacterium, what you can see is that there's, so Propionibacterium is a classic skin microbe, and you're probably seeing it a lot. Those of you who are looking in the indoor environment, seeing it a lot in those samples. So there's a whole lot of it everywhere on the face, except on the lips where it gets washed out by the oral community, and the ears where it's a little too cold for it to grow. And then if I show you Prevotella, which is a classic oral bacterium, you can see there's a ton of it on the lips, and then there's just a little bit of it in one of his ears, and then on one of his cheeks. And again, you can come up with your own scenario for how those oral bacteria might have got there, right? So, so by linking QIIME up to other tools, you can get this additional power to look at spatial patterning, and Antonio and Yoshiki especially have also been putting together a lot of time series tools that let us do more statistics on the time series data that's now starting to come in. So this might lead you to wonder where our microbes come from in the first place, and if you have dogs or kids, you probably have dark suspicions about that. And in fact, it does turn out to be true that dogs share more microbes with their owners specifically than with other people. I want to talk briefly about the data integration efforts. So I'm just going to mention that some work we did with Maria Gloria Dominguez looking at sampling the microbiota 20 minutes after birth, and then the mothers were swabbed an hour before they gave birth. Where what you can see, what you can see here is that vaginally delivered babies have communities that strongly resemble the vagina, whereas those that are delivered by C-section have communities that instead strongly resemble the skin. And here's another view of the same data with all of the body habitats from all the vaginally delivered babies clustering with the vaginal samples. And then over here, in light blue, all the body habitats from all the C-section babies clustering together with all of the skin samples from the mothers, and then the oral habitats are very different. Then we can also do things like look at development over time. So this is some work we did with Ruth Lay, where she traced one baby weekly over the course of the year. And so what you can see is the development of the microbiota in this very different state, finally coming to resemble the mother's community at day 838. And so what we can do is we can start to do things like integrate that with the Human Microbiome Project data. So Antonio and Yoshiki put together this visualization that basically allows you to ask how does that one infant's microbiota relate to everything that we saw in the Human Microbiome Project. And so what you can see is that the infant starts off, so this was a vaginally delivered infant, and so they start off strongly resembling the vaginal community. And then you start to see this very rapid change against this framework of the vagina, the skin, the mouth, and then stool samples. Again, using completely different, using different primers, and being integrated together through common metadata. And so what you can see is that the progress isn't very steady, right? There's a lot of back and forth. And so you have this period where you have the baby's microbiota bouncing around between the skin and vaginal community. Coming up in just a moment. Yeah. So this is when the baby takes a course of antibiotics, and you see this radical change, but then a relatively rapid shift back. And then by the time we get to the end of the time series, you can see that the baby's gut microbial community has started to resemble adult stool. So again, this kind of process where you take a reference frame of data, and then you take one time series and you animate it in that reference frame, can be extremely powerful for understanding what's going on, especially as in this case when you're combining samples from a lot of different habitats. Okay, so let me just quickly show you what you can do in the context of the QIIME database itself. And I am going to try to do this live, despite the internet limitations in this hotel that Mitch alluded to. So what I'm doing is just logging in here, and so you can see that currently we have a little over 26,000 samples in the database, a little over 2 billion sequences, and a fairly wide range of biomes. And so the database was primarily put together by Jess Dombauer, who's now at Thermo Fisher, Doug Wendell, and Gail Ackerman. And Doug and Gail are both here at the meeting and available for questions and things. And so you can do some things without logging in, like you can so you can look at where the samples came from, what biome they came from, and so forth. And also you can get a breakdown of where they are geographically. So we're starting to get pretty good coverage, although focused in North America, but from all over the world. So if I log in, Doug is going to demonstrate the process of actually uploading data tomorrow morning on a small test data set. So what I'm going to show you here is a meta-analysis capability. Assuming this page loads, which I realize is a bit of a risk in demonstrating this live. Well, so while I'm waiting for that to load, one thing you can do. So the get study summary and raw data link. Eventually takes you to a page where you can eventually takes you to a page where you can pick out any of those existing studies and pull it down. And so the idea is that you can get access both to the raw data and also to the process data. And if you click on the link for the QIIME database processing protocol, what it's got is it's got some information that describes to you how we do the standard processing for the database, and how we get all of these studies into one universe of OTUs. Let's see, Doug, you didn't get a chance to check this with the internet connection at the hotel, did you?
I uploaded this small test data set earlier, so it was pretty slow.
Okay. Well, I'm just trying to figure out. Am I going to be waiting for like a couple of minutes, or could this take 15 minutes to load or something?
The meta-analysis shouldn't take any additional time. It's just slow UI elements.
Okay, cool. Well, so I'll go to the meta-analysis then. And so, right, so possibly the next time we do one of these workshops, we'll pick a hotel that has a faster connection, if that's okay for it. Because, yeah. Okay, so what I'm going to do is I'm going to create a new meta-analysis. And what I'm going to do is I'm going to grab the data from that one baby that we were looking at, and then instead of showing it in the framework of the HMP data, what I'm going to do is I'm going to show it in the framework of a whole bunch of diverse mammals that we sequenced as part of another study. So this gives you an idea of how rapidly you can do a meta-analysis where you're combining two arbitrary data sets that happen to be in the database. And so this should load reasonably fast. Here we go. So as you can see, there's a fair number of studies in here. And if we take, so if we take the infantine series, and then we combine that with, for example, the human samples and the mammals from Microbial. I just need to wait for the rest of the user interface elements to load. So while I'm waiting for this to load, I'll just go through the balance of the slide. So basically the vision with the database is to allow you to see your samples collected in this universe of other samples. And for example, MGRAST has similar goals for metagenomic data. What we're focusing on at this point, although we do allow you to upload your metagenomic data to EBI via this mechanism, we're mostly focusing on the analysis and visualization of amplicon studies. And I'll elaborate on this more tomorrow, but it's really things like the Genomic Standards Consortium standards, like the mixed family of standards that Jack alluded to, where B marks is the minimum information about a marker gene study, for example, that allows us to take this data and integrate it in a consistent way. And so we're using these in things like the Earth Microbiome project, which is the source of a lot of the samples in that database. And having this large database lets you do a lot of additional cool things. So for example, with Scott Kelly, Dan Knights, who recently graduated from our lab, put together this very nice Bayesian source tracking method, where what you can do is you can take a whole set of entire community samples, And then for an unknown community, and so what we have is some different labs that we were swabbing for contamination, a neonatal intensive care unit, and then various things around offices. What we can do is we can take those habitats and we can ask how much of the how much of the community comes from each comes to the repertoire of each of a set of other habitats. So here the skin, the gut, the mouth, the skin, the soil, and so on. And so, for example, we can see right away that one of these phones has mostly soil microbes on it, whereas another one's almost entirely dominated by the skin. And so, so this is stuff that you can currently do directly with QIIME through the command line. We're in the process of figuring out how to do it on a larger scale via the database. So this is finally shown up, although it's not showing me any metadata. Doug, do you think this is just do you think I should just leave this to load, or should I reload it, or what?
We can try one more thing. I just turned the hotspot on my phone, which is about 10 times as fast as the hotel network. So you should try that. Do that.
Oh yeah, sure. Actually,
it's already on. If you just want to.
Yeah, sure. Let's do that. What's the
password? Just password.
Great.
I5 will show up as the network.
Great. Thanks. Yeah, it's a different connector, unfortunately, because of course they changed it. So just turn that on and off. There we go. Yeah, great, excellent. Okay, so let me reload that. Great, thanks. There we go. All right, so there we go. All right. Yes, so this is going a lot faster. Thanks, Doug. Okay, so let's see. So if we take the infant time series, and this time you can see that all the metadata is loading, and then if we take those mammals, grab all the so you can pick individual metadata fields, or you can just grab the whole lot, and you can also do things like sort or select by metadata. So for example, we could just pick a particular age range if we wanted to. And actually, I want to make sure that I get all of it, not just the common fields. Okay, and so if I continue there, what we can do is basically basically what we can do is we can do a whole lot of things like visualization with Topiary Explorer, which Meg Perrig wrote, and which shows you phylogenetic trees. You can do heat maps, taxonomy summaries, alpha and beta diversity plots. So if, for example, we just want to get to a 3D plot relatively rapidly, I can just click that and submit. And so what this is doing is it's running on our cluster. It's running on our cluster at Boulder, which we're expanding to make it more feasible to do this kind of thing for the increasing number of users that we currently have. And so what you can see is already it's produced the mapping file, the OTU table in this new format called Biome. And then a zip archive that just has those two files together. And so you can download the data at that stage for exporting to other programs for other analyses. It's also now doing the second stage, which is going from the data to, for example, 3D plots. And by default, it does unweighted and weighted unifrac, although there's a choice of about 50 other distance metrics you can use as well. So. So if I pull up one of the 3D plots, then go down and look at let's see, what did I do? Yeah, so basically. basically what you can do is get very rapidly to the get very rapidly to the kinds of analyses that you're used to seeing in QIIME. And so for example, here what we're looking at is the mother sample, which and looking at how the community is arched over to match that. What did I do wrong here? So let's see. Let me go back and rerun that, because I think what happened there is it only picked up. Only picked up the last one of those. Okay, so this time, if we select all of the metadata and all of the samples, and go forward with that, and go to the 3D plots. So what you can see is that it leaves on the meta. So you can do multiple successive meta-analyses, and you can clear them out at the end if you want to, or you can just. Okay, so I don't have an explanation for this. Let's go back to the beginning there. Obviously, when I practiced this before the meeting, it worked fine. Okay, so if I grab that one. All the metadata and go forward. Yeah, so that should all work. And we'll try that again. Doesn't like problems, which is surprising. Okay, okay. Well, rather than trying to troubleshoot this now, maybe I will investigate further this evening and repeat this bit tomorrow. But basically the point is that basically the point is that many of the things that you can do in QIIME from the command line, you can also do by grabbing arbitrary samples from the database and combining them into a meta-analysis like this. And so we've used this in a variety of environments. For example, you saw the bathroom data early on today. Gilbert has a follow-up paper on the kitchens, where again you can use these kinds of techniques to very rapidly both assess where particular kinds of microbes are, and also what the sources of those microbes are in terms of other environments you have in the reference database. And a few of you were interested in QIIME in the cloud, and so one thing I thought I'd ask. So we have a 3-hour block tomorrow designed to allow people to do some exploration of their own data. How many of you brought data sets that you would like to explore during that 3-hour period? Could I see a show of hands? 4, 5. Okay. So one thing that Greg and I were wondering is would it be more useful to do a demo of so for those of you who don't have your own data sets, which seems to be the majority of people sitting in the room, would it be useful to use this room to do a demo of running QIIME in the cloud using the IPython notebook? So that's a more graphical way of running QIIME than most of you have been used to. Would that be a good use of people's time? Could I see a show of hands as to how many people would be interested in that? Yeah. Okay. Great. So so we will do that tomorrow afternoon. For those of you who have your own data sets, what we'll do is we'll do a breakout session with some of the QIIME developers, so that you can sit down, try processing those, and so we can troubleshoot any problems that come up and so forth. And so in here we'll do the Greg will do the overall demo, looking at using IPython and using IPython and Amazon EC2, and then we'll do a separate breakout session for those of you with your own data sets. And I just want to point out that the cloud technology works really well in a wide range of settings. So, for example, earlier this year, Kathy and Luke and Antonio and I did a workshop in Bangladesh where even with a very limited internet connection, which is something that we can relate to here, and also where the power goes out frequently. So, for example, it's not possible to have a cluster on site because by the time it boots up, you'd have another power outage and have to reboot it. This kind of cloud technology can really make the techniques a lot more accessible because you no longer have to be responsible either for maintaining your own cluster or for begging the IT people at the university to give you access to a cluster that they might have access to, but that's more difficult for you to access. And so, so this kind of technology can be really empowering. So I'll wrap up with that. I'd like to thank the large number of people currently and formerly in my lab who've worked on both the data sets that I've shown you and especially on QIIME itself. So what you can download off GitHub as of Monday. We recently moved it from SourceForge. What you can download there is exactly the same as the version that we use in the lab. All of the features are checked in immediately as they're developed, and then as far as asking questions go, everyone puts in a huge amount of time on the QIIME forum to try to get your responses in 24 hours. So if you have a question, probably a lot of other people have the same question, and by using the forums, you can make those questions open to everyone. Another thing I'll mention is that a large part of QIIME's development has been working with different collaborators on different projects and different data sets, which is what allows us to make it relatively robust in a range of different systems and environments. And so part of the purpose of this meeting is to make sure that you know who the people in the group are who are developing QIIME, so you know who to talk to specifically if you're having a problem that you can't answer. So with that, I'd be delighted to take a few questions if there are any. Yeah.
The source tracker capability, is that, where you actually can portion it, is that now in QIIME?
Yes, that is in QIIME. So, and there's a whole lot of tutorials. So, for example, if you Google...
yes, the tutorials, we like those.
Yeah, so for example, let's see, I thought, was it tracking? I think there's a specific source tracking tutorial up there, although yeah, so either in the QIIME documentation or in the source tracker documentation, there's a fairly elaborate set of instructions on how to use it. So currently, what it's doing, so what it does is it takes a set of input communities, and then for each set of communities, it infers a Dirichlet model, and then what it's doing is it's partitioning your new communities among a model that trains on those existing communities plus an additional category for unknown. But yeah, that's all available and it's integrated